Poster titled Towards a Personalized Pharmacogenomics Workflow in Practice is a project related to how to analyze and integrate individual genotype data for pharmacological applications in disease treatment. Conventional therapeutic approaches are mainly based on a one-drug-fits-all model in which for a disease outcome, a general treatment such as a drug compound is prescribed to general population. However, for many diseases, including cancer, it has been observed that not all patients respond equally to the treatment. The same type of treatment can result in differences in efficacy and toxicity across patients. An approach to account for individual level variability is the use of pharmacogenomics. Pharmacogenomics is the study of the role of an individual's genome in drug response. Pharmacogenetic variation is common across populations, as an individual has at least one pharmacogene that describes him/her as a normal, slow, or fast metabolizer, leading to different effects on drug compounds. Thus, by integrating an individual's genotype data into treatment prescription, a clinician or pharmacist can predict what will be the potential response of a patient and therefore adjust dosage requirements or prescribe another type of compound by increasing drug efficacy and reduce adverse effects. The application of pharmacogenomics in clinics requires a computational automated toolbox to analyze genetic data. In addition, it needs to include readable, interpretable reports on drug interactions relevant to the patient's indication. For this endeavor, we aim to develop a prescriber-centric workflow that integrates bioinformatic tools, curated pharmacological databases, and personalized views such as graphical user interfaces or GUI to facilitate the use of a patient's genomic data when selecting treatment. We propose the following workflow as a potential application of pharmacogenomics. Starting with the request of a clinician or a pharmacist and the consent of the patient, genotype data is analyzed to identify the relevant patient pharmacogenes and alleles in gene-drug pathways. The main input of the workflow is a genotype dataset in variant calling format or BCF file. However, if the patient does not have an available BCF file, a sequencing or genotyping procedure is requested. The workflow includes all the necessary steps for processing DNA type datasets into BCF files, including read mapping, and variant calling for sequencing data, and quality control for hybridization array data. Once the BCF file is obtained, further quality control and annotation are applied to identify the significant variants in drug metabolizing pathways. Here, we include specialized tools for annotating pharmacogenes and star alleles describing metabolizing phenotypes. In addition, we include information on curated databases such as the Clinical Pharmacogenetics Implementation Consortium or CPIC guide, or the PharmJKB annotation database for clinical annotation and detecting gene-drug pathways, including the ones relevant for cancer treatment. A novelty in this step is by assigning a confidence level score on the query results based on the hits found in the gene-drug pathway analysis. In this way, clinicians and pharmacists can have a reference guide on the potential drug response of a patient and be more certain about how to prescribe the drug compound. Query results are then formatted into generalizable output for reporting, such as a GUI. A tracking history system is also implemented in the pipeline by collecting statistics in the overall analysis without retaining the patient genomic information. This system is set to ensure the reproducibility of results, monitor, and improve the performance of the toolbox. Query results as the main output will be mapped for reporting. In this step, we propose the use of Fast Healthcare Interoperability Resources Standard, or FHIR, to allow interoperability between healthcare systems. As for reporting, we propose the use of a main front-end interface that formats the query results into readable pharmacogenomics report, mainly for clinicians and/or pharmacists. The GUI will include the patient genotype information, gene-drug interactions and observation, and interpretation and recommendations. To ensure confidentiality and data regulations such as the GDPR guidelines, we enforce security protocols such as login credentials for only the clinicians, pharmacists, and our patients can access the data. The pharmacogenomics or PGS report will include information on the requester, and the patient. In addition, it highlights information on the pharmacogenomics tests, such as type of test, overall pipeline analysis, and institution that carried the test. As key features, our proposed workflow will allow genome data interpretation by identifying genetic variants of pharmacological relevance. The application will also provide a comprehensive analysis on how specific genetic variations may affect a patient's response to certain drugs. In addition, it will provide drug dosage recommendations aiming to optimize treatment efficacy and minimize side effects. Our application will also have the capability to interact with electronic health record systems to fetch relevant patient data and save pharmacogenomic results seamlessly. Finally, our expected outcome is to offer a healthcare system for personalized drug recommendations, aimed to improve treatments, outcomes, and increase patient satisfaction. With our workflow, we want to predict potential drug-gene interactions that will help to reduce the risk of adverse reactions, making treatments safer and more efficient. We also aim for the workflow to be also as an educational tool for healthcare providers and patients about the benefits and implications of pharmacogenomics and personalized medicine. We believe in engaging our end users through our design and development process to ensure our application truly meets their needs. Assembling a multidisciplinary team, including computational biologists, bioinformaticians, alongside with software engineers, UX, UI designers, geneticists, pharmacists, and medical professionals will be the key to the successful design and implementation of the PGS application in practice. This project is supported by the Flemish Institute of Technological Research and the Data Science Institute from the University of Hasselt. The project is funded by the Flemish Research Fund. Thank you very much for your attention.